Bismillah ar-Rahman ar-Rahim. This is the data show for the liver histological slides. As you see in this picture, this is the liver, and the most important thing is this portal vein, which is carrying the nutrients from the intestine, carrying the absorbed glucose, amino acids, glycerol, and fatty acids coming to the liver through this portal vein that will send branches in different parts of the liver. The liver, as any gland, is formed of stroma and parenchyma. The stroma is formed of the capsule. This is the capsule, which is called Glisson's capsule, which is characterized by being thin, formed of collagen fibers and the cells which secrete these collagen fibers, which are the fibrocytes. And it is covered, of course, by peritoneum, which will be in the form of simple squamous cells, but not evident here in this picture. From the undersurface of the capsule arise the connective tissue septa or trabeculi, and these trabeculi are actually very thin in the human liver, but thick in cases of fibrosis and normally thick in the liver of pig. The third component of the stroma is the reticular network or reticular formation, which is formed of reticular cells and reticular fibers, and it is stained by silver. So this is the reticular network of the liver, because here we can see the reticular fibers around the central vein and present inside the blood sinusoids. Parenchyma of the liver is formed of the hepatocytes that can be described in the form of classic hepatic lobule or portal lobule or liver acinus. Three ways of description of the hepatocytes arrangement. The classic hepatic lobule, it is hexagonal. The portal lobule is triangular. The liver acinus is diamond in shape. The classic hepatic lobule is formed of the hepatocytes that drain their blood into one central vein in the center of this lobule. It is demarcated by connective tissue, trabeculi, and at the corners of this lobule there is the portal tract or portal canal. In case of the portal lobule, it is formed of hepatocytes that drain their bile in one bile duct in the center of this triangle. The apices of the triangle are formed of central vein, and the triangle includes material or tissue from three hepatic classic lobules. The liver acinus is a mass of liver tissue surrounding a central vascular core. This is the central vascular core. This core is formed by branches arising at right angle from hepatic artery and portal vein. So this vascular core is formed by branches arising at right angle, زاوية قايمة, from the branches of hepatic artery and branches of portal vein present at the portal tract or canal. It is formed of parts of two adjacent classic hepatic lobules. So this portal lobule formed of parts of three classic hepatic lobules, but the liver acinus is formed of parts of two classic hepatic lobules. And it is divided into several zones. Zone 1, which is very near to the vascular core. Zone 2, in the middle. Zone 3, at the periphery of the liver acinus and towards the center of the classic hepatic lobule. This is the picture for the classic hepatic lobule. It is a hexagonal mass of hepatocytes, 6 sides, hexagonal, and these hepatocytes share with each other that they drain in one central vein in the center of this classic hepatic lobule. It is demarcated by the presence of this connective tissue septa or trabeculi, and at the corners of the lobule we have the portal tract or the portal canal, which is an area of connective tissue containing 4 important structures. A branch of the portal vein bringing the nutrients. So this one is carrying the nutrients which are absorbed from the small intestine. And a branch of hepatic artery bringing oxygen. And as well as we have here the bile duct, but we should remove this oxygen now. This is the bile duct, and it is characterized by the presence of simple cubical epithelium. That will be clear in other pictures. So this bile duct to drain the bile from the hepatocytes of the hepatic lobule. Also we have a lymph vessel for the lymph drainage. Lymph vessel for lymph drainage. Again, this is a picture of the classic hepatic lobule, hexagonal mass of hepatocytes that drain their venous blood into the central vein in the center and demarcated by connective tissue septa, which are very evident in the liver of pig, not evident in the liver of human. And at the corners, you can see the portal tract, but it is not evident here in this picture. So we have the central vein, we have the boundaries of the classic hepatic lobule, and this is the connective tissue septa, which is well developed in the liver of pig, ill developed in the normal human liver, but in case of fibrosis or cirrhosis, تليف الكبد, these septa will be well demarcated. Then, how can you identify the hepatic lobule in case of human liver, which has thin septa? We have three characters. First of all, you will see the central vein. Then you will see from the central vein these rays of hepatocytes radiating from this central vein. And then you will look at the periphery. You will notice that we have a connective tissue containing branches of the vessels, and this is the portal tract or portal canal. So this is a portal canal, this is a portal canal. Also here we have a portal canal, and maybe also in the other corners. This is a very nice picture for the portal tract. At the corners of the classic hepatic lobule, you can see four important structures. First of all, this is the branch of portal vein, which brings the nutrients. This branch brings the nutrients of portal vein, the nutrients, المواد الغذائية, which are absorbed from the small intestine. Number two, you will see a branch. This is a branch. Here, this is the branch of the hepatic artery. So we have also branch. Don't say hepatic artery, it's wrong. You should say branch of the hepatic artery, which brings the oxygen for the hepatocytes. Then we have here a lymph vessel, like a capillary lined by flat endothelium, flat squamous cells, and white inside, containing lymph, which is not stained here. So we have the lymph vessels. Number three, for the lymph drainage. So number three, we have lymph vessels for drainage of the lymph from the hepatocytes. Number four, here it is a very nice picture for the branch of the bile duct. Here, the fourth structure, here we have branch also of the bile duct. This bile duct, as you see, here it is lined by simple cubical epithelium. How do I know that it's simple cubical? From the shape of the nucleus and its location. It is central and rounded. It is one layer, so it is simple cubical epithelium. Okay, so these are the four structures, three branches, branch of portal vein, branch of hepatic artery, branch of bile duct, and we have a lymph vessel. Or you can remember them that we have vascular tissue, branch of portal vein, branch of hepatic artery, lymph vessel, and lymph drainage, which is a branch of the bile duct. Also look at the hepatocytes here. You can diagnose the presence of these RBCs. These RBCs are present in the blood sinusoid. So these red cells with the central rounded nuclei appeared as it is arranged in cords. These are the hepatocytes. And the spaces in between containing the RBCs, these are the blood sinusoids. So these are white spaces containing RBCs. These are, all of them are blood sinusoids. What is the origin of the blood in these blood sinusoids? It is the branch of the portal vein and the branch of the hepatic arteries. These are the sources for the blood present in the blood sinusoids. This is also a very nice picture for the hepatocytes. Here we have the central vein containing the blood drained from these hepatocytes. And these are the cords of the hepatocytes, acidophilic with central rounded nuclei, radiating appear as it is radiating from this central vein. And in between these cords we have these spaces containing RBCs. So these containing RBCs are the blood sinusoids. Look at this one, it's very nice blood sinusoid, and as it is opening in the central vein, it drains the blood finally to the central vein. So the blood sinusoids takes blood from the branches of hepatic artery, branch of portal vein in the portal tract, and the blood passes through this blood sinusoid to supply the liver cells. And the waste will be collected again in the blood sinusoid to be drained into the central vein. This is a very high power picture for the hepatocytes, showing the characteristics of these cells. We have here the hepatocyte. It is hexagonal, 1, 2, 3, 4, 5, or pentagonal, and they are acidophilic, and they having central rounded clear nucleus with prominent nucleolus. Why they are acidophilic? Because they contain a lot of mitochondria for energy and smooth endoplasmic reticulum for the cholesterol and phospholipid synthesis and the detoxification of drugs and hormones. We have here also the appearance of here this cell. This cell here, it is binucleated, and this is due to the high activity of the liver cells. And also you can see here the granules inside this cell and the vacuolation. You have vacuolated cytoplasm due to dissolved glycogen during preparation of the section. We have here also the blood sinusoids. This is one of the neutrophils here appearing in the blood sinusoid. So we have here blood sinusoids evident by the presence of the blood cells. Here and there, all these are blood sinusoids in between the cords of hepatocytes. If you want to see these glycogen granules that appear vacuolated in hematoxin, and you should use the PS stain or the Best carmine. PS stain will give you a purple or magenta color for this glycogen. All these magenta colored granules are glycogen granules. But with Best carmine, the granules will appear red. These granules are red in color, and these represent the glycogen granules present in the hepatocytes, and this is the nucleus, of course, and this is the hepatocyte, and these are the blood sinusoids. So if it is magenta color, so it is PS. If it is red color, it is Best carmine. This is a very nice picture to demonstrate the blood flow in case of the classic hepatic lobule. Here we have at the corners, we have branch of hepatic artery, branch of portal vein. They will pour their blood here into the blood sinusoids. And the blood sinusoid will supply the liver cells with the nutrients and oxygen and take the waste products and carbon dioxide. To pour this blood, which is now venous blood, into the central vein. So this is the flow of the blood from the periphery to the center of the classic hepatic lobule, where the central vein is present to drain this venous blood containing waste products and carbon dioxide. Look at this picture. Here we have the blood sinusoids. These are the spaces in between the cords of hepatocytes, and they are carrying the oxygenated blood with nutrients. Why? Because the oxygen is coming from branches of hepatic artery, while nutrients are coming from branches of portal vein, carrying these nutrients absorbed from the small intestine. Here we have the hepatocytes, and at the center, this central vein will drain the venous blood coming from the blood sinusoids after taking the oxygen and nutrients, and after exchanging the gases where carbon dioxide now is present in this venous blood, to pass to the central vein lined by flat endothelium, and then this central vein will carry the venous blood away from this classic hepatic lobule. Notice here the hepatocytes having a central rounded nucleus, clear with prominent nucleolus, and they are hexagonal or pentagonal in shape, and they appear vacuolated. Why we have vacuoles here? Due to dissolve of the glycogen present in huge amounts in this liver cell. Again, high power picture, very nice picture showing the blood sinusoid. Here the blood sinusoid containing the RBCs as well as white BCs. These are nucleated cells, and they are present between the cords of hepatocytes. All of these are blood sinusoids, as you see, and these are the hepatocytes, acidophilic cytoplasm, pale vesicular nucleus with prominent nucleolus. Some vacuoles appear due to dissolved glycogen during preparation of the section. This picture to demonstrate the lining of the blood sinusoid. As you see, this is the blood sinusoid, and this one also, and this one. Why? Because they contain the blood. Okay? Then, if you look to the lining wall, you can diagnose here this flat nucleus. So this is the nucleus of the simple squamous endothelium, which is forming the lining or the main lining of the blood sinusoid. So you cannot see all of the sinus of the lining cells. You can see one or two. This depending on the plane of the section, does it pass through the nucleus or not. Here in this sinusoid, you can see additional type of lining cell, which is the von Kupffer cell, which is a large cell. And of course, the nucleus is large. Relatively, this is a larger nucleus and this is a smaller nucleus. This is not flattened. It is a much irregular cell, a nucleus, but this is flat nucleus. This is the von Kupffer cell lining the wall of the blood sinusoid. In this condition, you may be asked about the origin of this cell. The origin is the monocyte. You may be asked about the EM picture. What is the most prominent organelle? Will be lysosomes. Why? Because it is a phagocytic cell. You may be asked about the function of this cell. It is the phagocytosis of all unwanted material passing through the blood sinusoid to prevent their arrival to the circulation of the liver or to the liver cells itself. So this cell, it is responsible for phagocytosis of unwanted material or any microorganisms present inside the blood in the blood sinusoid. So if you are asked about the EM picture, or the most important organelle will be lysosomes, and you will see also inside the cell phagocytic particles which have been phagocytosed by this cell. If you are asked about the stain, it should be stained by vital stain. Why vital? Because this stain is injected inside the living organism, and after being phagocytosed by this cell, the organism is killed and dissected, and the section is prepared to see the pigment inside these cells only. All the phagocytic cells are stained by vital stain, like trypan blue or India ink. What is the function? As we have said, it is the clearing of the blood and phagocytosis of the bacteria and microorganisms in this blood, in addition to the phagocytosis of all the RBCs. They are in close relation to the RBCs here, so they can phagocytose all the RBCs, and that will help in the formation of the bile pigment. If you are asked about what is this red line, it is the cell membrane of the hepatocyte. It is the side or the face facing the adjacent cell. So we are expecting in this side to have junctional complex. What you are expecting to find here in this surface facing the blood sinusoid, we are expecting to have very long microvilli for the absorption and for the exchange with the blood inside this blood sinusoid. So finally, the blood sinusoids are lined mainly by the endothelial cells and also with the von Kupffer phagocytic cell. Here we have stained the section with a vital stain. The stain is injected in the living organism, and it is left for some time for the phagocytosis takes place, and then it is dissected, and the section for the liver is prepared to demonstrate these von Kupffer cells. And also it is stained with hematoxin eosin, as you see the section, but this stain applied was the vital stain to stain the von Kupffer cell. And then after this section and taking the section is stained by hematoxin and eosin. You will see here that the stain, the vital stain, the color of the vital stain, either it is trypan blue or India ink, is present only in these cells, in these von Kupffer cells, inside the blood sinusoid, and it is not present at all in the liver cells, but only in the blood sinusoids, in the von Kupffer cell. So this is a good demonstration for the function of the von Kupffer cell. Here also we have the central vein lined by the endothelial cells, and the blood sinusoid is opening inside this central vein. And the liver cells, acidophilic cytoplasm, and it is vacuolated due to the presence of glycogen that dissolved during preparation of the section. This is a very nice picture to demonstrate the von Kupffer cells by vital stain. Here the stain is called India ink, and it is phagocytosed during the living state by the von Kupffer cells. And after that, the animal is dissected, section of the liver is prepared and stained by hematoxin and eosin, so that you can see that the hematoxin and eosin stained the ordinary hepatocytes, but these von Kupffer cells are stained with the India ink, which is a vital stain. Here all these are the hepatocytes, vacuolated cytoplasm, acidophilic cytoplasm, vesicular central rounded nucleus, and it is hexagonal or pentagonal in shape. This is a very high power to demonstrate a very important structure or space called space of Disse. Here we have the hepatocytes, acidophilic cytoplasm, vacuolated and may be binucleated, and the nucleus is vesicular nucleus with prominent nucleolus, as it is an active nucleus in an active cell. But here you will see these are the blood sinusoids, lined by the von Kupffer cell, which is a large branched cell. This is the nucleus of the von Kupffer cell here, as it is originating from a monocyte, so it has more or less a large irregular nucleus. And here we have a space, a very narrow space, between the blood sinusoid and the hepatocyte. This is the hepatocyte nucleus and this is the cytoplasm. This is called the means around, sinusoidal space. Here it is evident in this location as this white line between the wall of the blood sinusoid lined by von Kupffer and the cell membrane of hepatocyte. This is the first start of the lymphatic tissue here, as it contains 4 structures. What are these 4 components in this space of this? First of all, we have the very long microvilli. No, I will make it in the other direction. These are the hepatocytes, so this is the hepatocyte, are projecting very long microvilli towards this space. So this is the first component. The second component, we have the plasma oozing from the blood sinusoid. So we have here plasma coming from the fenestrated blood sinusoid. Here we have plasma passing through the fenestry of the blood sinusoid to this space. Number 3, you will have reticular fibers forming a reticulum here in this space. These fine fibers branching, forming reticulum to support the wall of blood sinusoid. So we have here, if you want to put them in their place, we have here these reticular fibers supporting the wall of the blood sinusoid. The fourth structure, which is a cell called cytocell or lipo, means lipid, lipocytes. Lipocytes, cells containing lipid inside. These cells are actually branching cells and containing droplets of fat. These droplets contain the vitamin A, which is a fat-soluble vitamin stored in these lipid drops. So this perisinusoidal space, peri means around, حول sinusoid, perisinusoidal space or space of this, the scientist who discovered it. It is the space between the hepatocytes and the wall of blood sinusoid. Hepatocyte here, wall of blood sinusoid here. And it contains the long microvilli of the hepatocyte to increase the surface area for the exchange between the hepatocyte and the plasma oozing from the blood sinusoid, and the reticular fibers supporting this wall of blood sinusoid and the plasma oozing itself. So microvilli, plasma, reticular fibers, and finally we have also in this space the lipocytes storing the vitamin A inside these lipid droplets in the form of retinyl ester. Retinyl ester inside these lipid droplets. This is an electron microscopic picture. If you look to this picture, you will diagnose the RBCs. These are the RBCs present in the blood sinusoid. So, this cell, it is in the lining of the blood sinusoid. This is the von Kupffer cell. This is a part of the nucleus appearing here, according to the plane of the sectioning, and it is full of phagocytosed particles inside this cell. This is, and of course, you expect to have a lot of lysosomes and phagocytosed particles, as we have said before. Then you will look to this big cell with a clear eochromatic nucleus. Here, the eochromatin is more than the heterochromatin, so this is 100% it is the nucleus of the liver cell hepatocyte. And you will look at it, you will find plenty of mitochondria. Also, we have other organelles, as this will be the rough endoplasmic reticulum near to the nucleus, and towards the periphery you can diagnose also smooth endoplasmic reticulum, but it is of a small magnification here. And you will notice also here they have microvilli, how these microvilli are projecting towards the wall of the blood sinusoid. So this is the wall of the blood sinusoid. These are the microvilli, mitochondria, cytoplasm, nucleus. Then look at this cell, which is full of moderate density droplets. Those are lipid droplets. So this cell is the I2 cell. Where is the location? It is in the space of Disse, in the perisinusoidal space between the hepatocyte and the wall of the blood sinusoid. Also, this is another hepatocyte because of the presence of the mitochondria, and we have here projecting microvilli towards the blood sinusoid. In this picture, you have a diagram demonstrating the bile canaliculi, which are present between the rows of the liver cells in the hepatic cord, between the two rows of hepatocytes forming hepatic cord. We have the bile canaliculi. It has no lining, no specific lining. It is enclosed between the hepatocytes. Till we reach toward the periphery of the classic hepatic lobule, you will have here the canal of Herring. This is canal of Herring, simple cubicle. Why cubicle? Central rounded nuclei, cubicle cells, towards the periphery of the hepatic lobule. They receive the bile from the canaliculi and drain to the bigger duct which is present in the portal tract, which is called the bile duct or the interlobular bile duct. Inter ما بين lobular ما بين اللوبيولز, between the classic hepatic lobules, it is present in the portal canal. So we have bile canaliculus enclosed between the rows of the liver cells, and then canal of Herring at the periphery of the hepatic lobule, and they will collect together to form the bile duct present in the portal canal or portal tract, lined also by simple cubical epithelium. And this one will collect from each portal tract to form right and left hepatic ducts. In this picture, you will see that this liver cell full of the mitochondria here, and it shows towards the side of the bile canalicus short microvilli. These microvilli are short, and the microvilli towards the blood sinusoid are long. This picture I have added to demonstrate the direction of the flow of the blood and the bile in the classic hepatic lobule. In this one here, you will see that in the portal tracts we have a branch of hepatic artery, this one, and branch of hepatic portal vein. These two vessels will pour their blood into the blood sinusoid. It will pass from the periphery towards the center where the central vein is located. Here the blood at the start is full of oxygen and nutrients, and as it goes towards the center it becomes venous blood, devoid of the nutrients and full of carbon dioxide instead of the oxygen. And this central vein will drain this venous blood to the outside of the liver. But here, in case of the bile passes between the hepatocytes in the space. The bile will pass in the bile canaliculus here, which has no separate lining. It is present between the two rows of liver cells. Towards the periphery, it will form a canal of Herring, and then these will be collected together from here and there to form this bile duct, which is present in the portal tract. So we have this one, this one, this one, this one, this one. All of these are lined by simple cubical epithelium, as well as the canal of Herring also is lined by simple cubical epithelium. If we look to this picture here, you will find that, I will make this small, you will find that. Here is a more magnified diagram. You will see that we have here at the portal tract or portal canal, branch of hepatic artery, branch of portal vein. They pour their blood into the blood sinusoids, and then this blood at the center is collected by the central vein. And then the central vein will collect this venous blood to what is called the interlobular veins, to hepatic vein that will pass from the porta hepatis. So this is the drain of the venous blood. This is the arterial blood coming towards the center. But here, in case of the bile, look at this. Here the bile coming from the center to the periphery and collect in the bile duct, which receives the bile from the canal of Herring, and with this receive the bile from the bile canaliculus and canal of Herring and the bile duct in the portal tract, and then this bile duct will collect together to form right and left hepatic ducts.